Our next talk is by Professor Chen, and he's going to be talking on growth hormone profile and influence on metabolic factors. After training as an MBBS at Fudan University in 1982, Professor Chen completed his PhD in neuroscience at the University of Bordeaux in France in 1989. He then worked in Glaxo Incorporated in the USA and headed the endocrine cell biology at the Prince Henry Institute in Melbourne until 2008. His work focused on the regulation of growth hormone, and he has published more than 120 papers in international scientific journals. He is currently NHMRC Principal Research Fellow, Professor and Chair of Endocrinology, School of Biomedical Sciences at the University of Queensland. Professor Chen served as chair of many international conferences and has delivered the opening plenary lecture at the 2010 A5M conference in Melbourne last year, and is also a fellow board member. Welcome, Professor Chen.
Thank you, Nathan. Thank you very much. Sure, I noticed that's handling up the time for the presentation. I will certainly try to make within the time. So what I'm going to tell you is just one part of the experiment we do in the lab. So I will focus on the growth hormone. So in my presentation, I'm going to go through those parts and try to convince you the growth hormone is probably the most hard-working hormone in our body, and it certainly has the link with aging and also with the link to the energy and to the metabolic disorders. And I will more focused to tell you a little bit about our research, especially the growth hormone profile, because it's a pulsatile secretion in the mouse animal model, and you all realize it's a very small animal. So for the pulsatile analysis, then you need to continuously collect the blood samples. So whenever we're talking about growth hormone, I mean, the first thing come to us is it's linked with growth. So it definitely show in those pictures I just got from the internet, some with excessive growth hormone, acromegaly, or really, really tall figures, and some very, very small, they don't have any growth hormone inside. So growth hormone truly linked with our growth, but they are not only stopped there. And the growth hormone has been used by many bodybuilders, and this is the picture I pick up from the internet. You can see this is 1982, and this is 2008, almost 30 years difference. And you don't really see too much difference in the muscles. And that reminds me a couple of years ago when I was in Melbourne, and I got a phone call from Sydney, and they caught him styling in the airport, and he got a big luggage box with all the human growth hormone, and its amount of hormone level is so high, which is far more than anyone can imagine. So certainly he's using a lot of growth hormone, and hormones certainly change the body composition. You can say it's almost keeps the muscles, and it certainly do a lot of things on the performance. And this is 1988, the Olympic game. You probably all remember the first one, Johnson, and he being caught by using the steroid, and the gold medal has been taken off. But one thing I think they didn't really report is, I look at that, it said he actually spent about $60,000 on growth hormone. So he certainly also used growth hormone. But whether growth hormone truly helped him, I don't think so. I mean, all research currently showing it's not really doing anything for performance. But you can say for the athlete, they do use a lot of drugs, and number one, number two, and a lot of them all have drugs. And the poor guy, the last one, they say, everybody uses drugs. It's a interesting thing. So growth hormone has been used for the people to build the muscles and to help them perform, but in fact that's not really help them to perform sports. But one thing truly happen is they do reduce the fat and they do increase the muscles. Those are the agricultural use of the chemicals to stimulate growth hormone, and that will help to produce the low-fat meat. So come back to us, our medical research, those are the growth hormone. It's a 192 amino acid. It's a relatively big protein, and it works on the growth hormone receptor, which is a cytokine receptor. It's a single transmembrane domain receptor. So growth hormone, where it comes from, it's actually come from the pituitary. I pick up this from the textbook that just to show you that the anterior pituitary gland, it's actually come from different part when we compare with the brain and the neural system. And they are not really come from the neuronal system, and they don't really link with the brain or the hypothalamus. And it only goes through with the portal blood circulation to regulate it. So it's anterior pituitary gland. Although it's just underneath the brain, it's a totally endocrine system, not a brain one. This is anterior pituitary gland. So they receive all the regulatory hormones from hypothalamus, goes through this portal blood circulation and released in the anterior pituitary gland, then regulates the hormones. So there are quite a few different hormones. You just heard about the thyroid, and this is growth hormone is also one of them from the anterior pituitary gland. So when we do research, as I just said, there are more than 5 types of endocrine cells. Then how can we know when we work on the single cells, are they a growth hormone cells? So what we do is we use a transgenic mouse. So what we insert is a GFP, that's a green color protein, and with the same promoter for the growth hormone. So every growth hormone cells, they will show this green color, and then we can work on them with the confidence we are working on a growth hormone cells because we do a lot of single cell study. And in addition to growth hormone, we have similar strategy for growth hormone releasing hormone that's linked with GFP, so we know they are the GHRH neurons. So when we're working on the hypothalamus, you see those are the GHRH neurons. They are all green color and median eminence with all the terminals to end to release a growth hormone releasing hormone. So growth hormone, they do have a lot of function, as I just said, not only for the growth and also with the function of the liver, the bone, the muscle, and also using the fat to provide energy. And the growth hormone releasing pattern is a pulsatile secretion. This is critically important. It's not only to keep a high level of growth hormone to produce any physiological function, it has to be pulsatile. In fact, if you compare the man and the woman, actually female's growth hormone level is higher than the man's one. But the only thing is our pulse is higher in the reach to the top. So that's how growth hormone produces a function, and pulsatile pattern is so important. And this is a brief diagram to show you growth hormone in the pituitary gland. It's regulated by releasing hormone, and also the other one we haven't touched on is growth hormone inhibitory hormone or the somatostatin. So that will inhibit growth hormone. Only these two hormones working together with the pulse of the releasing hormone, and when the inhibitory hormone is going down, then you have a pulse. If you have an inhibitory hormone here, even you have releasing hormone, you won't see a pulse. So it's a very close regulated pattern to regulate the growth hormone secretion. So the central regulation of the release of the it's from the hypothalamus, and the two principal regulator I just mentioned to you is the releasing hormone and somatostatin. Both compounds produced and released by the neuronal population located in the hypothalamus, goes through the portal blood circulation, reaches the anterior pituitary gland. So growth hormone, one of the major one regulated by growth hormone is IGF-1. I think most of you all know this. But in addition to stimulate the liver to produce IGF-1, growth hormone receptors do exist in bones, in muscles, and fat. They do have their own effect without IGF-1. So IGF-1 is not the only one to responsible for the growth hormone's function. And they have feedback regulation to regulate the growth hormone secretion. And what we currently moving from here is to say what is the metabolic regulators and how they regulate the growth hormone secretion. So this is growth hormone positive secretion in human, and you can say it's clearly this is man, this is woman, and you have clear positive secretion, and this positive secretion is linking with the human function. So when we try to study that in the animal models, the mouse is the one obviously have a lot of advantage because we have so many transgenic knockout, and we can study all the factors of hormone, particularly in relation to their functional metabolic regulation. But the growth hormone secretion in mouse is so hard to measure just because it's too small. So what we do is we first thinking about how we can develop a method we can actually measure the growth hormone pulse in mouse. So what we collect the blood is from the tip of the thigh, and then we collect only 2 to 4 microliters whole blood for growth hormone assay. So one thing we need to go over, or one hurdle we need to go over is to leave the animal without stress and to collect the blood samples and timely with every 10 minutes and go for up to 6 hours. So they don't really feel anything at the back, and you can collect the blood samples and they don't really feel it, and you press a little bit, then you stop the blood and put them back to the cage, and then whenever you have a cotton close to them, they will go into this paper towel and paper towel centers, and then you can collect the blood next time. So this is the one we can collect blood samples, and we can collect them from the free moving without stress, because stress will wipe out all the growth hormone pulse in all animals. Then the next hurdle we have to go over is a highly sensitive one. Normally for growth hormone assay, we do the assay from the serum, and the serum, you know, its amount is very small, and basically using that technique, you just can't get enough blood to do the growth hormone assay. So that's why looking through the publication is mostly one time point, collect blood samples. And so what we do is we can go down to very, very sensitive. I'm not going to go through this too much into details, but what we can do is we can collect the samples and do the growth hormone assay in full blood without separating the serum or the plasma, and then we can correct the volume and to get the growth hormone levels in the plasma. And the sensitivity can go down to we only need 2 microliters of blood. So if we collect 4 microliters for double measurement and we do 6 hours, the whole blood we collected is less than 8% of the whole blood volume in the mouse, which is still within the range of the NIH regulation for one blood sample collection from the animal. So it's a highly sensitive and doable technique. That's the whole process we go through to establish the method, but it's all in press now in endocrinology, will be published pretty soon. So this is the growth hormone pulse from the mouse. You can say we have very clear pulse, and if we fasting them for 12 or 18 hours, then we say their pulse is disappeared, and very clearly disappeared. Even you go with this, the y-axis to go in a high range, and you still don't see. You do see a little bit of the pulse, but it's far less than the normal pulse. So they are sensitive and a doable technique for the growth hormone, I see. So positive growth hormone secretion, and it's a clearance observed the hyposomatotropism of the obesity in man. So in man, you can see this is controlled, this is obese, and you can see the growth hormone pulse is certainly reduced, and the amplitude is reduced as well. So growth hormone deficiency does occur in human, and many of the research showing that. And this is the other human study. So you can say with the high-fat feeding for 3 days or 2 weeks, you can say their growth hormone pulse is all disappeared, and that's the normal growth hormone pulse. So growth hormone have a very sensitive link with the metabolic balance, and they do change along the different metabolic conditions. So this is our experiment model. So we feed the mouse with a high-fat feed from the 4 weeks of the age. This is very early compared with the other high-fat feeding experiment. And you can see slowly the body weight does going up compared with the control. And this body weight increase combined with the growth hormone reduce. And this is 1 dot points we collected. And we also see the ghrelin level going down and leptin level going up. This has all been well established in the research when you have the high-fat feedings. And also the free fat acid, fat tissue is going up. So what happened to their growth hormone pulse? This is the one thing we currently try to do, and we want to see whether they do have a reduction in positive growth hormone secretion, very likely. So this is the other experiment. They show the growth hormone regulation of lipolysis, and in the other term is this growth hormone pulse. It's all depending on the GHI-RH pulse. This is the one I just tell you. The growth hormone secretion is stimulated by growth hormone releasing hormone. So if you use growth hormone releasing hormone antagonist, then you wipe out all the pulse secretion, and same thing for this one. And this is during the early fasting period. In human, fasting or reducing feedings certainly stimulates the growth hormone pulse. And this is different to mouse. I think for mouse, probably because their size is too small, they don't have enough energy to be stimulated by the growth hormone. That's why their growth hormone secretion is reduced until the latest stage. If you do energy restricted feeding for a long time, then growth hormone level will go up. So this is the other the fat levels. You can see this is 12 hours fasting or 2 days fasting. But if you have the growth hormone releasing hormone antagonist, then you reduce this free fat levels. And this is one of the evidence people showing to say the growth hormone increase in human in fasting is actually stimulate the free fatty acid secretion or release into the blood circulation. But in mouse, it's very different. So growth hormone in human, what we can say is it's actually sprouts the protein at the expense of the fat, using the fat to provide energy and to protect the muscle and the proteins. So those the one I showed you before. This is fasting for longer time, and you still 18 hours. You do say they are still no growth hormone secretion. We currently try to do is using the energy restricted, not the fasting, because the fasting ethically we cannot do too long. Actually, 18 hours is quite long, and we can only do the energy restricted feedings, and hopefully we will see the growth hormone pulse coming back later. So you can see this is one, the free fatty acid in mouse is going up very, very quickly. And from 2 hours and until 18 hours, it's certainly still up. So this is the mouse which is different to humans. So we show that human, we got growth hormone increase and then the free fatty acid increase. In mouse, it's actually free fatty acid going up much faster, even without growth hormone increase. And this is the onset of fasting on the glucose. You can see the glucose level is very quickly dropping. And we are currently working on the insulin, and hopefully maybe they have more link to the insulin glucose levels, and then to try to use fat to provide energy. Okay, this is the growth hormone and the fasting. Those are the pulse. This is the fasting time, so the growth hormone is not going up, but free fatty acid is going up. And the reason for that, we're still working on that, and it doesn't seem to be the growth hormone stimulated fatty acid. That's similar results I just showed you. And IGF-1, you can say one thing we're thinking about is whether this is because IGF-1 going up and negative feedback to reduce growth hormone. It doesn't seem to be the case. And you can see the IGF-1 is dropping in the fasting mouse. So that means the IGF-1 reduction is more the consequence of the low growth hormone level rather to cause the low growth hormone. So the other thing I bring into this topic is the ghrelins or the GOAT or acetyltransferase. You probably know ghrelin is a hormone which discovered to release growth hormone. And this hormone comes from the stomach, and it has to be acetyl to be active. And those enzymes actually makes the non-active ghrelin to active ghrelin. So this enzyme has been shown to be important for keeping the animal if they have energy restrict. So that's the most surviving parts of the growth hormone. You can see this one. So this is the blood glucose levels in this knockout animals. Those are the control. And then the plasma ghrelin, active ghrelin, certainly going up in wild type, but you don't see that in the goat. And similarly, you see a lot of diacetyl ghrelin or the inactive ghrelin, and compared with wild type, it's probably even more than the wild type, but you don't see any active ghrelin. So those animals, we are thinking about whether they have anything to do with the growth hormone pulse, and we are using this animal in the future to study growth hormone pulse. So hopefully that will tell us whether ghrelin play a role in the fasting or the energy-restricted feeding, and how they regulate the growth hormone pulse, and then how they play a role in the aging process. So those are the probably the taking home message. What we say is a versatile and valuable research tool is using the mouse to study growth hormone pulse, just because we have so many different genetic modified animals we can use to target on certain hormones or the certain factors. And it can do a detailed analysis as well, just because we do have the tool, we can study that. But whenever we do this study, then we need to carefully consider the difference between mouse and human. Apparently, mouse are different in certain part, and in the other part can be similar. And one key element we need to think about is the mouse is an animal. They never stop growing, and their growth hormone probably play a role all life to keep them to go up and bigger and bigger. Not like humans, after 20 years we won't go any further. The other thing is the mouse size is so small, so the metabolic rate is so quick, and their response is much faster compared with the humans. And does that fit to the human condition? That's always the things we need to consider whenever we do research or do the data analysis and try to apply that to the clinic situation. But mouse, as I just said, is very small, so it's very difficult to do the experiment. But, I mean, probably there is no limit to what we can do, but what we are only hindered by our imagination. If we can thinking about, I mean, this is just because when I started to do the growth hormone assay from mouse, and I talked to many people and the endocrine colleagues, they said it's impossible. It's just too small, and the blood level is too small. But we actually can do that. So we have a lab in University of Queensland. So for growth hormone assay, that's the one I reported today to you. And hopefully, if you have any chemical or compound you want to say whether they have anti-aging or probably correct the endocrine growth hormone deficiency, we can certainly work on that and help you to go through the collaborative work. And in addition to growth hormone, we're also working on the ALS dysfunction in diabetes. We work on ischemic heart disease, and especially with the small molecular peptide, we are hoping to develop to treat the ischemic heart disease. And we have a small project working on the endometrial cancers with a link to the local ghrelin and ghrelin receptor and how they be involved in the cancer proliferation and cancer growth. So that's the group. I mean, this is actually last year's photos. Quite a few people go and a few people come. And those are the 3 people mainly involved in this growth hormone experiment. With Derek is a postdoc, and Lily and John is a PhD student in the lab. Thank you very much. That's my presentation. Thank you.